Okay, hello to all of you. I'm going to start giving you instructions for the next lab we're going to do. So basically, we are going to start a mini project, and in this mini project, there will be three labs. In the first lab, we are going to basically isolate the proteins from the spinach, and in the second lab, we are going to purify that proteins because when we isolate the proteins, it's not a pure prep. There are many other proteins, but we are specifically interested in a specific protein, and that protein is called Rubisco. And in the last lab, we will be running the protein gel. We will be doing protein gel electrophoresis, and we will be able to see our desired protein. So basically, yeah, I hope all of you know what proteins, and we will be working on a specific protein called Rubisco. And Rubisco is a enzyme basically which is involved in photosynthesis. So in the Kelvin cycle, there is the first step where carbon dioxide goes and combines with a compound called RUBP, and then it forms a compound which is having six carbons, and then the whole Kelvin cycle starts in photosynthesis. So we will be basically in this mini project, we will be basically isolating Rubisco. That is our first lab. In the second lab, we will be purifying Rubisco from other, we will be separating Rubisco from other proteins. And in the third one, we are going to, we will be running our fractions we will be keeping on the gel, and we will be looking for Rubisco in the gel. Okay. So here is some information about Rubisco, as I've already told you. An important thing to know here is Rubisco has two major polypeptide subunits with molecular weight of approximately 55 kilodaltons and 14 kilodaltons. So this is the plan. This is a mini project which we are going to do for 3 weeks. Week 1, we isolate proteins by ammonium sulfate precipitation. Week 2, we do ion exchange chromatography to purify our protein. And third is protein electrophoresis. So for isolation of the protein, we will be using a technique called ammonium sulfate precipitation. Now, in the first lab, we are using ammonium acetate precipitation. So we must know how this ammonium sulfate precipitation works and how it separates the proteins. So we will be actually adding, you will be see that, so you will be doing in the lab today, on Monday and Thursday. Basically, we will be adding sodium sulfate in increments. We will be increasing it, and with the help of that, the proteins will be precipitated. Now, how the proteins get precipitated? So basically, like these proteins, they're interacting with water molecules. When we add the salt starts making interactions with the water, and then the proteins, they precipitate out. So basically the salt will go and make the interactions with water, and the proteins will not be making interactions with water, and they will just precipitate out, and that's what we want. So basically, this technique manipulates the solubility of proteins by replacing polar interactions that the water has with the proteins and forcing the water to instead interact with the salt ions. In a high enough salt concentration, the salt ions will interact with and displace all the water molecules interacting with the protein. When the protein is no longer participating in hydrogen bonding with water, it then precipitates. This precipitate can then be collected as a pellet following centrifugation of the sample. So that's what we are going to do in the first lab. So let's go over the protocol. Okay. So what you're going to do as the whole class, we will be giving you spinach, and each group will derib approximately 50 grams of fresh spinach and blot dry with paper towels. And then what are you going to do? So every group is going to do that. And then collect the spinach leaves from the class. So all of you will be collecting this deribbed spinach leaves, which is like around 50 grams every group is going to do. And then all of you are going to put them together in a blender, and you're going to homogenize them in approximately 200 ml of buffer I. So the buffer I will be provided to you, and the what is there in the buffer I is given here. but it will be ready made for you. You will be just using it. So basically you are going to collect the leaves from the whole class, and together you're going to add 200 ml of buffer I, and then you are going to blend it in the blender, and basically you're going to make kind of a smoothie. Following homogenization of the solution, the solution will be divided and distributed to each group. So then this smoothie kind of a thing which is made, which is like a spinach smoothie, you will be dividing this thing among your groups. And then what are you going to do? Filter the resulting suspension through 2 layers of Mira cloth. So this cloth will be given to you. You have to just filter it and then estimate the volume of the filtrate. So you have to estimate how much filtrate you have. And out of this filtrate, you have to just save 1 ml of the filtrate. And you can just save this in a microcentrifuge tube, which will be provided to you. And this you're saving it because we will be running this filtrate on the clean gel during the third. So now you have saved filtrate and have the left. So the calculate the amount of sodium sulfate you will be adding, and you have to calculate the amount of sodium sulfate so that you reach up to 37% saturation. And to reach to 37% saturation, you need actually 210 grams of 1 liter of solution. So if you have one smoothie or a homogenate, you need to add 210 grams of ammonium sulfate to reach to 37% saturation. But you do not have 1 liter of the solution. You will have either 40 ml or 50 ml. According to that, whatever the volume you have to do a calculation. And this is the formula. And whatever you have, 40, 50, whatever, you do the calculation. And according to that, you need to figure out how much ammonium sulfate you have to add. Then very slowly add the solid ammonium sulfate using a spatula while stirring the filtrate on a stir plate. So you will be provided with stir plates, and you will be having your smoothie in the beaker, and slowly you will be adding your ammonium sulfate. So you have to first calculate and then add slowly, and on the stir plate it will keep on moving, it will keep on stirring. It's very important that it should be stirring throughout. So once the ammonium sulfate has been added, continue to stir for 15 minutes. You have to do this thing for 15 minutes. After 15 minutes, pour the contents from above step into 50 ml centrifuge tube. So that same falcon tubes which were given to you in the last lab during the subcell lab, you will be taking all the contents from your beaker into 50 ml centrifuge tube. Then the tubes from all the groups have to be balanced. So we will be again going the way we did last week. We will be going to the centrifuge. I will help you with that. And every group will bring their tubes, and we are going to centrifuge for 15 minutes at 9000 G at 4 degrees Celsius. Okay, we'll be doing the spin together for 15 minutes at this much is the speed. So after the centrifugation will be done, you will see a pellet at the bottom and the supernatant. Now what you have to do is you have to take that supernatant and put into a beaker. Okay, so you have a pellet in the centrifuge tube, and you have the supernatant, and you're going to transfer the supernatant to the beaker. And out of that supernatant, you're again going to save 1 ml of the supernatant, and you're going to label your tube as S1, and you're going to put it in the fridge. I will be giving you boxes, and you will be giving me all your fractions, and we will be putting them in the fridge, because all these fractions we will be saving throughout. We will be requiring during the week 3rd. Okay, so now estimate the volume remaining in the beaker. Again, we have to do the ammonium sulfate precipitation, but this time we will be doing 50% ammonium sulfate precipitation. So we started with 37%, now we're going to 50%. So what we are doing, we are increasing the amount of ammonium sulfate in increments, and we are trying to precipitate the proteins. I have already explained the principle that proteins were making bonds, they were interacting with water, but as we are adding this salt, now those interactions are being disrupted, and the water is actually making the interactions with the or bonds with the salt, and the protein is getting separated. So you will again do the estimation by using this formula. So to reach up to 50% saturation, you need to add 85 grams of ammonium sulfate into the 1 liter of the supernatant. So using this formula, whatever the supernatant you have, calculate it, and then you will be doing the same kind of stirring you have done in the last step. So you will be putting your the supernatant on the stir plate, and you will be adding the ammonium sulfate slowly in increments, and you will be stirring it for 15 minutes. Okay. Now what to do with this? This is what we did with the supernatant. We saved some, and the leftover supernatant, we have done the 50% ammonium sulfate precipitation. What to do with the pellet? Now read the pellet in get a second pellet. The actually, yeah. Yeah, this is, sorry, this is with pellet 1 only. Yeah, you will be suspending the pellet in 4 to 7 ml of the water, and if your pellet is big, you can increase the amount of water too. You can go up to 8 ml or 9 ml. And then what you have to do, you will be given some dialysis tubes and the clips. I will help you with that. Then we will be adding your dissolved pellet in water to the dialysis tubes, and then we will be doing the dialysis against the buffer A for 2 days, and I will take care of your dialysis for 2 days. And basically what we are doing in dialysis is, we are actually, in the pellets there is a lot of salt, because remember that we are using ammonium sulfate, which is a salt. So we are precipitating we are precipitating the protein, but there will be some salt too. So we will be doing dialysis so that we can remove the salt from our protein prep, from our isolated protein. So dialysis tubes, basically they have some pores in them, and the size of the pore is such that it will let the salts go out of the bag, and it will retain the proteins inside the bag. So we will be doing dialysis for 2 days, and I will keep changing your buffer, so that all the salts which are there inside the tube, inside the dialysis bag, they will go out and the protein will stay inside. Okay, so this is with pellet 1. Okay, now you are stirring your supernatant 1 for 15 minutes. You have done that with the 50% saturation of ammonium sulfate. So after that is done, we are again going to centrifuge the tubes together for the whole glass at 9000 G for 15 minutes. Okay. Now this will be so reaching to this point, we have started dialysis with P1, and we have saved some S1, and we have also done we have also added the ammonium sulfate, and we have done the stirring, and now we are going to spin them. So after spinning, you will see again there will be pellet at the bottom, and then there will be supernatant, and that supernatant is S2. Okay, so following centrifugation, transfer 3 to 5 ml of the supernatant into 15 ml conical tube labeled as S2, and put aside for protein electrophoresis. Dissolve the pellet in 4 to 7 ml of distilled water, just the way we did for P1. Label it as P2. Then again gather dialysis tubing and clips. Place the second pellet suspension into a separate dialysis bag, and dialyze against buffer A at 4 degrees for 2 days, changing the buffer in between. Okay. So after dialysis, P1 and P2 will be further purified based on the differences in formal negative charge by ion exchange chromatography. So ion exchange chromatography will be done during the week 2 lab, and it is basically for purification of our protein. So we will talk about how purification is done through ion exchange chromatography. That is for the next week. So reaching to this point, you have saved F1, sorry, the filtrate or F1. You have saved S1, S2, and we will be storing them in fridge. And your P1 and P2, they will be in the dialysis bags, and they will be getting dialyzed for 2 days. I will take care. And once that is done, we will them dialysis and evaluation. Going for discussion among yourselves.